Good afternoon, everyone. We're glad that you're here today for our next presentation in the EMSL LEARN Webinar Series. My name is Genoa Blankenship, and today we're going to be talking about computing resources for mass spectrometry. But first, we wanted to remind you that next month we will be having another EMSL LEARN webinar, and the topic will be focused on top-down and native proteomics. It's scheduled for Wednesday, December 8th at noon, and we hope you'll join us. Also, just a couple of other items. We wanted to encourage you to subscribe to EMSL's email list. We're going to drop the link in the chat so that you can add your name and your email address if you haven't already. And also wanted to encourage you to be following EMSL on our social media platforms, LinkedIn, Twitter, and Facebook. But today I wanted to introduce you to our three speakers. EMSL computational scientist Yuri Corilo will be giving us an overview of the open source data analysis software suites research area. He's also going to be discussing the development of a new mass spectrometry software framework called CoreMS. After Yuri's presentation, we're going to hear from Will Q, who will be discussing embracing ambiguity and complex mixture mass spectrometry analysis. And then we'll end with Lisa Bramer, who is a data scientist with PNNL. She'll be discussing Freda, a framework which allows users to upload and explore mass spectrometry. We will leave time at the end of today's webinar for questions. We encourage you to post your questions in the Zoom chat, and then we'll have all of our speakers answer them during the Q&A. With that, I'd like to welcome Yuri.
Hello, my name is Yuri Corilo, and I'm a computational scientist here at the Environmental Molecular Science Laboratory. So today we're going to briefly discuss the development of a new mass spectrometry software framework called CoreMS. The framework was developed for small molecules and complex mixture analysis. Working with me on this project, we have a great and diverse team from chemists, data scientists, software engineers, IT engineers, and computational scientists. Software development for modern mass spectrometry analysis is a complex interdisciplinary endeavor, which requires skills in computational sciences, chemistry, physics, and a deep understanding of the mass spectrometry techniques. So, to enable the scientific community to keep pace with these fast improvements in the mass spectrometry technology, we have developed a Python software framework that's called CoreMS. There are three main components of the software framework. The first one, it standardizes the molecular identification workflows by merging state-of-the-art approaches and the first principle of mass spectrometry. The second, it established a modern, stable, and sustainable software framework by providing the building blocks for the data processing pipelines, independent of the platform that you're using. The third, it provides the tools for one to build acquisition time data processing platforms that facilitate the data, metadata capture, and at the same time, it provides a reliable. So the first step towards achieving our goal of consolidating the software development was to create a standardized, modular, intuitive mass spectrometric data structure that is represented here in the middle of the slide. You can see that different data types are represented by different object types, and they are organized, the relationship of those data are organized in a hierarchical manner. So if you have an LCMS experiment, you have a collection of mass spectrum. So when you parse the data from the raw data of the vendor through the CoreMS framework, you will have an LCMS representation of the data. Inside the LCMS representation of the data, you have a collection of the mass spectrum objects. Inside the mass spectrum objects, you have a collection of mass spectrum peak objects that can then have further information about the molecular formula depending on the level of the molecular characterization you have done. So with the structure of the software defined, we move to the development of the data processing workflows. So we worked on the development of a new and novel direct infusion FGACR workflow. We also have a GCMS metabolomics workflow. We're actively developing the CMS metabolomics workflow. We are also working on centralize the access to the molecular reference data by development our architecture that will allow us to seamlessly communicate with the framework through APIs. So the first example of workflow we're going to show you today is an example of direct infusion, high resolution mass spectrometry applied for the characterization of complex mixtures. So the first step towards achieving that was to move us away from a static molecular formula reference database. So the software now has this dynamic molecular reference generation that's based on your mass spec data. We also made sure to include support for all the ion types, closed shell, open shell, including metals and halogenated compounds. We have an automatic fine isotopic structure calculation that is based on the monoisotopic assignment, but it also used the dynamic range to accurately calculate what are the isotopologies you should see on your data. We also created a new novel confidence scoring mechanism of all the putative molecular formula assignment that's based on two components, the mass accuracy and the spectral similarity between the calculated fine isotopic structure and the observed isotopology on your data. So the second example is LCMS data-dependent acquisition of 18 metabolite molecular standard compounds. So here on the center, you can see the total ion chromatogram and all the track ion chromatograms and the possible features identified for all the targeted ion in the protonated form. In the zoom in set, you can see the feature that was selected and highlighted for further investigation for the cinnamic acid with a rotation time of 7 minutes. So looking further into the feature, we access then the MS1 level information acquired, and then you can see that the software automatically recognizes what the search space should be for that specific peak feature, chromatographic peak feature, and the software gives you all the details of the molecular formula assignment, the mass accuracy, and the confidence score alongside with the isotopologic score. So you can see the identification of the monoisotopic and the identification of the isotopology. So if you have more than one option of molecular formula within the search space, the software will give options and then it will select the most confident based on the confidence score. So the software then automatically access the dependent scans, and if the window is within the range of error for that specific M over Z, the software will again automatically select the search space and then allow for the assignment of all the M over Z's into the MS2 mass spectrum from the parent ion to the fragment ion. So you have the molecular formula for all the features that you see, and in addition to that, the software automatically calculates the cumulative neutral loss based on that molecular formula. So you can clearly see here an accurate representation of the CO loss and the CO2 loss for the cinnamon. Because the CoreMS framework allows for the fast development of new molecular identification workflows, here I'm showing you an example of a GCMS-based metabolomics workflow that was developed in a matter of days. All the signal processing and the molecular annotation methodology was based on previous work published called metabolite detector. We did, however, improve the M/Z-based deconvolution by adding a hierarchical clustering of the peaks that gives us a better representation of the reconstructed mass spectrum. We are also working closely with Chavian and Lisa to establish an improved similarity score that's based on rotation index and the spectral similarity. So there are multiple level access for the framework. The base code for the framework is available at the GitHub EMSL Computing repository. And there's also two command line interface for common workflows like the direct infusion analysis of complex mixture by FGICR. We call it VAR-MS. There is also a command line interface for MetaMS, data processing of GCMS, where we will in the near future add the capabilities for the LCMS. Right now, if you want access to the LCMS code, there is a development branch on the CoreMS framework. We also have a graphic interface, is a web application. We will also be making that public available in the near future. That anyone can just get a code base and reproduce and have a graphic interface to be able to use the software to process the data. And that can be deployed easily using Docker Compose. And I'll talk more about how we decide to deploy this inside EMSL. So here on EMSL, we decided to deploy the stack using Kubernetes. We have a cluster we call the Razor cluster, and all the services here was built thinking on microservices. So every part of the software that does a specific function, it has a specific container for that function. So usually a user goes and it uploads the data, goes into the portal, uploads the data is then stored in the cloud storage setup. All the metadata is then stored on a Postgres database. So the user goes back to the portal, select the data, select what the type of workflow he wants to process, and then that request is then submitted to the Freda database. We are using Celery here to manage the job queue, and we have multiple runners. Every runner is a specific workflow, a LCMS workflow, a GCMS workflow, a direct infusion workflow. That way we can distribute the data processing across different nodes, or it can even be deployed on personal laptops. So the data processing happens on the runner side. The runners automatically communicate with the centralized molecular reference database, where all the reference data is in silico and the molecular standard data lives on the centralized database. Then the results are deposited on the Postgres, and the user can then go back and look at the results on the web portal. So one of the limitations of the architecture I showed you on the previous slide was that the user has to manually upload the data to the portal to be able to process their data. So what we're working now is in the functionality of automatically upload the data at acquisition time. So on your acquisition station, you're going to have a new software that automatically recognizes the new data is generated. It will automatically upload that data to the mini.io cloud storage setup, and then automatically process the data and show you the results on the web. Right now, we're working on a project that would do the QC for the FGICRs. We are using Surfa against Suwannee River fulvic acid as the QC. And then we have a dashboard of statistical importance results that the operator can check on data quality. This gives us, moving forward, the ability to have consistent results across different studies by consolidate the data processing pipeline, and also gives us the opportunity to automate the instrument parameterization by looking at the QC result and automatically changing the instrument if something goes wrong. So I hope I was able to show you today that with the creation of the CoreMS framework and the analysis portal and the ability of acquisition time data processing, that we have the foundation for the standardization of the mass spectrometric based workflows, while also ensuring a consistent and reliable stream of data products and data in metadata capture. So thank you so much for your time today. I would like to acknowledge some colleagues here at EMSL, David Colley from the EMSL Molecular Science Computing Group, Ian Smith from the PNL Research Computing for their help and support to this project, Thermo Brooker for providing us the library to access the raw data. Next up today, we're going to have William Kiel, and he will expand on the idea of embracing the complexity and the ambiguous molecular formula assignment. It's a work that it's embedded on the core algorithm of the molecular formula assignment, the CoreMS, and we strongly believe it's really important that this work will enable sample agnostic molecular formula assignment. And we're also going to have Lisa Bramer, and she's going to talk about Frida. That is a web application that was developed to enhance data exploration and data visualization for direct infusion experiments of complex mixture analysis.
Hi there. So yeah, I'm Will, and I'm going to be talking today about how we can use CoreMS and this new framework to embrace ambiguity when we're performing complex mixture analysis. I don't have control of the screen, Laura, sorry. So there we go. Oh, somebody's gone too fast. Thank you. There we go. Okay. The path of what Zoom webinars never went smoothly, I don't think. Let's see. Okay, so what I want to talk about is complex mixture workflows, specifically when we're talking about doing soil organic matter characterization. So you might go out into the field, perform some experiments, do some sample preparation, and then we'll do some data collection using high resolution mass spectrometry, such as Orbitraps or ICR. And then we perform data analysis. And this is where CoreMS and Frida comes in. And so I want to highlight today the formula assignment aspect of the data analysis workflow. And then after me, Lisa will talk about statistical analysis aspects with Frida. And of course, I'd be remiss not to mention that EMSL as a user facility will enable you, if you collaborate with us, to perform many of these things using our resources. So please get in touch if you want to collaborate on any of your experiments. So formula assignment is not a solved problem. There are a number of papers that have come out over the last 15 years or so about how to do analysis of electrospray ionization, complex mixtures. And these include research from our lab, such as Formularity. There's a sort of standout paper in 2006 from Kuczynski lab showing about how you can perform these automated analyses, workflows, but even new papers coming out in the last year or so. So it's clearly, there's a lot of opportunities out there. There's a lot of tools. But what I want to stress is that all of these tools tend to be built on the same principles and the same, heuristics and legacy opinions. And why are we building CoreMS if we already have all of these other tools? Well, one of the main things is that it's not just a formula assignment tool. So as Yuri said, it is a framework for data analysis. So you're able to go from raw vendor data through to processed annotated data in one pipeline, in one interface, which makes it more reproducible, it makes it high throughput, and it also makes it more auditable. You don't lose metadata or anything at any point. You don't have to convert between file formats. Being open source Python code, it's flexible, it's customizable. If you have some custom algorithm you want to implement, you can do so. Also, if you don't necessarily understand how our algorithms work, you can look at the code and make sense of them for yourselves. But fundamentally, it presents an opportunity for new ways of thinking, for new paradigms. We don't have to think the way we did 20, 30 years ago. And you're able to experiment with these ways. And one of the key things that we're excited about is the ability for data-informed processing. So as I said, formula assignment is not a solved problem, potentially controversial opinion, but I think if anyone thinks about it rigorously, they will agree with me on that. And the implication is not just there. It means that any downstream analysis, which relies on unambiguous formula assignments and assumes all are equally true, is therefore going to be biased towards the limitations of the upfront analysis. So some of these common assumptions that come into play in the legacy tools include the idea that unambiguous formula assignment is possible, and it's built upon a number of principles which we know are not true or not necessarily true. Sometimes these things can have validity, but oftentimes you make an assumption up front before you actually look at the data. So for example, people assume that complex mixture ESI spectra are full of singly charged ions, but we know that that's not true, and there was even a paper in the last year that proved this. There are the assumption that in negative mode, they're all deprotonated ion types. There's assumptions about elemental constraints and mass accuracy. And the fundamental one that gets me is this idea that one peak is one formula and not more. And I can demonstrate in some slides later on how this is not true. So as an example of using CoreMS to perform a formula assignment, here's a data set we acquired relatively recently on an older Orbitrap instrument. So this is a high resolution mass spectrometer, but it's about 10 years old. And so we can get high resolution of about 100,000 with M/Z400, and we get a nice complex mixture. There's several strong features per nominal mass, as well as a number of smaller GRAS features. With CoreMS, we're able to read that raw data directly in, perform a peak picking function, and then without having to worry about doing anything else, we can just do a wide formula search to try and understand the nature of the data without going in with too many biases. So in this case, we use a massive formula error tolerance space of ±5 ppm, acknowledging that the instrument should be within those constraints. We also allow for deprotonated ions and chloride adduct ions, and we allow for CHON chemical elemental space. And with this formula search, we see a clear distribution of features in the center of this error plot. We can determine, therefore, the distribution of errors that are present, and then CoreMS has built into it a robust recalibration function, which is actually largely automated. And knowing the prior distribution, we can therefore recalibrate it and get much nicer mass error without having to manually inspect every spectrum. Recalibrated, we're then able to, this is so slow. Sorry. Recalibrated, we're able to refine our formula search. So we can use much tighter error tolerances now that we know what the actual error distribution is. We can include the elemental formula that we'd allowed before, but we can actually expand that space. And so we go from having about 1,800 assignments with 300 ambiguous to 2,500 assignments with about 400 of these remaining ambiguous. So the question is, how do we deal with them? Well, legacy tools use some arbitrary rules, which again, sometimes are true. These include the using the peak with the smallest mass error. So if one peak could be multiple formula, arbitrarily picking the one with the smallest error or the smallest number of heteroatoms, or in fact performing the search but allowing fewer elemental constraints. So for example, most tools don't necessarily allow you to search for different ion types at the same time. And a lot of people won't include phosphorus in their assignment search because it's pretty uncommon to get confident phosphorus assignments in many of these samples. Well, CoreMS allows a more open-minded way of thinking about it. So we have both the confidence score, and we're supporting the idea that you should embrace ambiguity. So as Yuri said, the confidence score is built off of a combination of the mass error score of the monoisotopic formula, as well as the isotopolog similarity for those monoisotopic features. So what it does is we're assuming that the mass error should be centered around 0, but we're allowing for a distribution. And with the isotopolog search space, as Yuri said, we take the signal to noise and the dynamic range of the spectra, and we work out what isotope features should be detected, and then we score them against the presence or absence of those features. And this allows us to get much more idea of quantifying how much confidence or evidence there is to support a molecular formula assignment. It is not to say that a confidence of 1 means it's true, it's just to say it's more confident than one that has a confidence lower. With this, we can downsample our data to only the most confident ones, and therefore we can actually go back to those original assumptions that people routinely make, and we can investigate those from the point of what is true in our data. So one assumption is that all ions are deprotonated, and here we have evidence that most of the ions are deprotonated, but there are a number of real adduct ion types here. Similarly, people would assume that the NSP count is low, and if you had a conflicting assignment, you would exclude the ones with these other heteroatoms. But we can see here, whilst these numbers are relatively low, there are real nitrogen, sulfur, and phosphorus containing assignments in this data set. So, sorry. When we have all of these ambiguous assignments, what do we do with that? We ultimately have to accept it's okay. Much like if you had two up balances in a lab, one with 2 decimal places, one with 4 decimal places, there's only a certain amount of confidence you can have with your measurement. And that is true with mass spectrometry. With the instrument abilities, dynamic range, with the data quality and the sample complexity, there is always going to be some ambiguity in your formula assignments. And there's a bunch of other options you could do to try and address this. You might use a higher performance instrument. You might revise your analysis using complementary sample information. You could use some orthogonal techniques to confirm or deny certain elemental constraints. You might decide to exclude ambiguous results, which is sometimes done. However, we're generally against throwing away data. Or you might decide to incorporate the ambiguity into your models. For example, if you're building an ecological model, including these different formula assignments for the same feature, but including with them some degree of uncertainty. And just very quickly, I want to highlight that with a few other data sets of higher resolution instruments, we still have this issue of ambiguous assignments. So here we're comparing two sort of cutting edge commercially available mass spectrometers, an Orbitrap from Thermo and a new FTICR from Bruker. We're looking at swan over fulvic acid, which is well characterized. And you can see that running these instruments at close to their maximum performance, we're able to get 12,000 features in the Orbitrap and about 23,000 features with the FTICR. If we limit our search space to CHO only, we get about 3,000 or so unambiguous so-called formula assignments. But if we expand that search space to include nitrogen, sulfur, phosphorus, and chloride, we get far more assignments in total, up to 18,000 assignments with the ICR data, but we still only have about 3,500 unambiguous ones. So this is despite the fact we've got a much more powerful instrument than we had with the Velos on the previous slides. We still don't have absolute confidence in most of our assignments. And one thing I want to stress here is this idea that one peak is one formula is obviously incorrect. If you look at the bottom panel of this figure, you can see that the Orbitrap data shows one feature in the center there, but the ICR reveals that that one peak was actually 3, perhaps 4 different features that the Orbitrap simply could not resolve. So if you go into your analysis assuming that every peak is perfectly resolved, that is probably most of the time an erroneous assumption. So the other thing that's great about the confidence score filtering is that we can get rid of features that we've assigned, and then we can look at what was removed and we can support that it was logical to remove it. So for example, assigning all of these features in the ICR dataset, we get this region of Van Crevelen space in the lower left, which is sometimes seen when we do these complex mixtures and allow for NSP. But if we look at what is removed and we compare it to the recent paper from Jeff Hawkes' lab, where they discovered that most of these features tend to be multiply charged ions, which are incorrectly assigned, we can see that using our sort of blinded, but just based on objective metrics, i.e. confidence, we're getting rid of features which are probably incorrect. And so therefore the confidence score is working well for us in this instance. Okay, and then finally, I want to show the way, even with the top of the line instrument, the 21 Tesla FTICR here that we have at EMSL, you can look at some complex mixture and you can make your measurements using broadband measurements, as we routinely do, or using this selective ion monitoring or quadrupole isolation, or however you want to call it. And with this, we can improve our dynamic range, we can record higher resolution measurements, and yet we still have these features which are incredibly close together in space, such as these two features, which I've annotated here. There are unambiguous assignments within our search space, such as the major peak that's just CHO, but there are ambiguous assignments. There's also features which are not cleanly resolved, even with a 21 Tesla FTICR. And so many of the assumptions that we're thinking about shouldn't be made even when we're using the best instruments that are available to us today. So to sort of summarize where I'm going, for complex mixtures, uncertainty always exists. And I think for too long, we as a community have sort of tried to strive towards complete confidence and certainty. And the fact of the matter is that we will never have enough resolution and sensitivity to confidently annotate every feature beyond doubt. That's just not how direct infusion ESI works. And so what we need the community to do is therefore understand the limitations, integrate complementary data, try and avoid as many arbitrary heuristics as possible, and use data-informed processing to embrace ambiguity. And then finally, just to remind you, CoreMS is not just another formula tool. In addition to the data-informed processing and the confidence scoring, we support multiple assignments. We've got this dynamic library generation. The algorithm is generalized, and this is a really key point. It means that this will work on organic matter samples, but it will work on proteomic samples. It will work on metabolomic samples. It should work on any type of complex mass spec data because it isn't built off of arbitrary heuristics in the past. And then it has also got this programmable interface. And then finally, many people at EMSL and PNNL to thank, primarily, of course, Yuri for building most of CoreMS, and then all my other collaborators here for either supporting in the instruments or providing data to present here today. And with that, I will hand over to Elisa, who is going to talk about Frida and statistical analysis.
Okay, thanks, Will. So Yuri and his team have built this really great tool of CoreMS that can handle all of these data. And part of our goal was to take downstream, as a data scientist, part of our goal is to take downstream identification of peaks and help users make sense of those. And so I'm going to talk a little bit today about a tool that we developed called Freda. And so just to give you back to the workflow that we're looking at, you've run your samples, done the data analysis, processed the signals, done formula assignment, and now you're ready to hopefully do some sort of statistics or make sense of your data. So with FTICR data, some common visualizations, which both Yuri and Will nicely alluded to, were Van Crevlin diagram and these Kendrick mass and defect plots. And a lot of times we just take a spreadsheet and we make one plot after another, which is not particularly efficient. So rather than that, my team is currently in the process of building a suite of tools that are called Multi-Omics Analysis Portal. And in the meantime, while we're getting that ready to roll out, we have this app that we call Frida, and that app is built on an R package that's open source, and you can find it at the GitHub location here, with the primary goal being able to take FTMS, so Fourier transform mass spec data, and trying to make sense out of it and do some of these calculations that have perhaps traditionally been done within spreadsheets and do them in a more automated and reproducible way and calculate attributes about the different peaks that you're seeing. So if you are more inclined to the programming side of things, this package is available. But if you're not programming inclined and you prefer to have a graphical user interface, that's what we've developed here, is this tool, which we've shortened to Frida, but really just stands for exploratory data analysis of these high resolution mass spectrometry data. It is R and Shiny based, and it's open source, and it's got that R package underneath that's driving things under the hood. So we go from what I have listed here as upload and format, but what we're currently working on is a seamless transition so that if you've run your data through CoreMS, and Yuri talked about the back-end architecture that's going on, we have similar architecture built behind Frida where we're deployed onto the Kubernetes Razor cluster, and I'll provide the link to where this app exists at the end of my slides here, but the goal then is as soon as you're done from CoreMS and you're satisfied with what you have, we can transfer that over to Frida App seamlessly, or you can continue looking at the data and perhaps calculate some characteristics about the different peaks you've identified, do some informed and reproducible filtering and data quality checking, and then producing some interactive visualizations and even making some statistical comparisons. So I'm just going to go through some screen shares here and talk through them. This is what the app looks like when you first come to it, and what you'll see is we have a section that has an introduction, some data requirements, and so there are example data sets there as well if you want to try this on your own or see what the format of the data looks like. Right now, the upload capability is the only way to get your data in here, but within the next month or so, you will be able to directly port from CoreMS and not have to deal with uploading CSVs. So what's happening right now on this page is we're just going through and specifying a few things like what column specifies peaks and where the molecular formulas are in the spreadsheet. And we tell it to process the data. And at the end, then it shows us how many peaks are in our data and how many samples we have in our data set. And if we move on to I might be stuck in an infinite loop here. Going from one page to playing a video. There we go. Okay. But one too far. Okay, well, the app also gives you the ability to specify samples and what types of groups that they belong to. So if you have different treatments you want to compare, and once you've done that and you move on, you can calculate some characteristics about the peak, like Kendrick mass and defect, or Gibbs free energy, nominal state, and that's potential energy, right? Nominal oxidation state of carbon. Once you ask that to process the data, what happens is the app will give you some summary statistics about those variables that you've computed. It'll also show you a plot of any of these variables, and then you can see here I've clicked on the top to ask it to save this plot for later, and then on the little folder I can see which plots are being saved for download for later as well. So then next we come to a quality control page, and this is really giving us a preview of our data, our samples, and here you're seeing how many peaks were observed for each sample. This would give you some insight into if there are particular samples with really a low number of identified peaks, anything that looks out of the ordinary. Additionally, you can always also get an idea of what the actual intensity measurements look like on a per sample basis. And here I've stored different groups of samples, and you can see I can click down to those particular groups of interest and get the resulting plot to really start to inspect these things as well. This works similar to what you've seen before, where I can save the image that as soon as I'm happy with it up in the top right with the little download icon. And once we feel good about the data that we have, we can move on to a filter tab. And there are a number of different filters. So at the very top, you can see we have sample filters, and that would allow you to remove anything you saw on the QC page that looked really, really odd and may influence your downstream analysis. In addition to that, we know that for these instruments, there are certain M/Z or mass ranges where we trust the instrument. We have the instrument calibrated, and so we allow users to put in those values depending on the instrument as well. And what you can see as soon as I've checked that filter on the right, the bar plot shows me how many samples were removed, the before and after of that. And so another filter that we often apply are things that are sparsely observed in our data set. So here I'm applying a filter that's taking out any peaks that were observed in just one sample, saying it needs to be observed in at least 2 samples. You can see we also have a place to do custom filters here as well. So any of those characteristics that we've calculated about our peaks, let's say that you have a great interest in, peaks only. In this case, I'm selecting one particular variable, NOSC, and if perchance you were only really interested in things like peaks that are have a NOSC greater than 0, this app will let you filter down to those particular peaks and downstream continue to do your investigation. And the great thing about this is at the bottom of this page, there is a reset filters stage, so that if there are certain subsets of the data you're really interested in, you can filter them, then come back, reset those filters, and look at a different subsection of your data. So after we're happy with the filters that we've applied, we tell the app and then move on to visualization. So visualization, we have 2 options. One is that you can just create some standard plots. The other is that we have what are called linked plots, and I'll just show an example of each of those here. You can see there's a download or drop down that shows many different types of plot options, including the standard Van Krevelen and Kendrick, but there are also density, custom scatter plots, and dimension reduction techniques. And so here I'm showing making a Van Krevelen plot for a single sample, and it will pop up with all the samples that I have to choose from. And once I'm happy with my selections, click on generate plot. It will on the fly generate the Van Krevelen plot for that particular sample. And we have options down at the left over here, like taking out, these are approximate boundary sets to classify the types of peaks. I can do away with that. Color by other aspects or characteristics of that I've calculated. So here again, if let's say I wanted to color by the Kendrick defect to start to get an idea. We can make density plots, which is what I'm doing here, and that's for each single sample or multiple samples, I can look at what does my distribution of NOSC values look like across that. And I think potentially then what ends up being potentially more useful is a Van Krevelen plot or other plots, but let's say that I want to actually compare 2 groups of samples. And here I think am selecting a comparison of 2 groups. So here I'm comparing lower native samples and lower upper soil samples. So these are soil samples that are native to one region of an area and samples that got transplanted. And what you can see here is rather than arbitrarily determining, we're trying to determine what peaks are unique to each set of samples or each group. Rather than arbitrarily setting thresholds, we can actually do formal statistical tests, in this case a g-test, to ask for what peaks are things uniquely observed in one group. That is, I see the proportion of samples that observe that those peaks is statistically significantly different than the proportion of samples that observe the same peak in my other group. So rather than drawing arbitrary thresholds, we then can get a resulting plot that looks like this, where the purple shows things that are observed in both groups. And then we have an interactive plot where on the right, I can click those purple points off and get an idea of what is actually unique to one group versus the other. So as I click that button off in this video, you can see that in the lower upper samples, they tend to be much higher in oxygen to carbon ratio than the native samples. So we can start to get insights about the data by doing these types of statistical comparisons where we're actually running statistics instead of determining arbitrary thresholds. And the last thing that I want to show here is dimension reduction. So PCOA is just based on presence absence rather than the actual peak intensities. But you can see we do that dimension reduction, you get a plot, and then that has cover capabilities where you can start to see the samples, and I can start to click on the different grouping variables that I created, and the plot then will update and color my points by so here are my lower samples, so samples that started in the lower region of the mountain versus upper. And I think in the interest of time, there are other graphical capabilities, but in the interest of time, we'll skip ahead and wrap things up here. So just really quick, one thing I want to say is that in addition to all of the visualization options, we have options to map things to databases like KEGG or Metapsych, and additionally you have the option to restrict which things you want to try to match to those databases. So rather than some of these formulas, as you know, because we don't know structure, are going to match to multiple compounds. So if you aren't interested in the things where there are 20 potential matches to your formula, you don't have to receive that data. You can set that threshold and get appropriate information about the different reactions and pathways that these peaks are observed with. And finally, we do have a download page. And what you'll find there is you can get all of your processed and filtered data, as well as the report of what was done to your data, and your database mapping tables. And you're also seeing on the left here all the figures that I've saved along the way, which I can eventually here click on. If I don't remember what those plots looked like, it will show me a preview of each of those. And I have the ability to then, say, never mind, I didn't actually want that plot. Go ahead and remove it from the list. Once I'm happy with my output, you get a bundled up package of everything that you've done. So just to wrap this up, I will say we are continuously developing this capability, and the direct connection to CoreMS is coming very soon. If you want to give this a try with some example data or data of your own that fits the current acceptable formats, this is the website that you can go to, and I'm happy to. My email is at the beginning of these slides and in the chat here, and I'm happy to entertain feature requests too. We're trying to make this as applicable and usable to folks across the board. So thank you very much.
Okay, with that, I think we're going to jump into some questions here. And Lisa, I'm going to keep you on. First question, is there a definition for the CoreMS output to allow data to move from additional data analysis tools into Freda?
So there is a standard format that data comes out of CoreMS, and it comes out in terms of files that are 1 per sample, and that contains all the aspects that Yuri and Will are talking about, right? The potential formula assignment, confidence metric, all of those things, peak intensity. And so right now it is, I will say it's not trivial to take CoreMS output and put it into Freda, which is why we're making this direct connection. And so the ability to take a stack of CoreMS files and process them into a form that Freda can accept will be available both via that R package that I showed at the beginning if you're code inclined. Otherwise, there will be just a mechanism either via upload or via the direct link with CoreMS where a user can do that without having to do all the data munging themselves. So that should be coming within the next month or so here.
Okay. All right. We have a question for Yuri. Yuri, you may have to revisit your slides here, but the question was on slide 9, there was a time course for MZ147 that shows multiple peaks. Was this simply a replicate data with a bit of time shift in the separation between runs, or a chromatographic anomaly of a single run?
So I think that's neither. That plot is the strike ion chromatogram. So, just for clarification, we have a target, list of metabolites and the strike ion chromatogram. So that's what that plot is showing you. So it is expected to see multiple peaks. And another thing is that this is Orbitrap data with a 5 ppm, air range for the strike ion chromatogram. So.
Okay. All right. Thank you. A question for Will. Will, do you account for the presence of homologous series during scoring? It can be hard to detect isotopologues or low abundance peaks, presumably penalizing them in scoring. Membership in such a series arguably should improve the score.
So yes, that's a really good point. We don't directly include homologous series in our confidence scoring because that would bias it to the assumption that you have homologous series. Now, that assumption is true a lot of the time for petroleomics type data, for really degraded organic matter like Swanner River fulvic acid. But sometimes we're working with fresh organic matter, so leaf litter extracts or something, which may have far fewer big repeating series throughout the molecular space. And so what I would suggest is that you could incorporate that into an additional algorithm, but that would be workflow dependent. And what we've tried to do with the confidence score is make it ambivalent towards the type of data that you're looking at and as universal and objective as possible. But you're also right that the isotopologues are hard to detect. And that is something that you have to accept. That is a limitation of the instrumentation. And so you shouldn't, features that are low signal to noise inherently are going to be less confident in their assignments.
All right. Lisa, are you considering a binary upload file format to keep the data file size reasonable?
We hadn't considered that. We haven't, to date, we haven't really run into this issue because this is running, the app itself is running on a Razor cluster. So the only trouble that we tend to run into is if you have, more than 100 samples, let's say, that's been run on the 21 Tesla, then at that point, we have some features built into Frida that will take away some of the dynamic plotting options to process things more quickly. But at that point, for a user, we would recommend the Shiny app itself is also available on GitHub, and we would recommend potentially just running it locally at that point. But so it hasn't been much of an issue to date, but I would certainly entertain that issue if it does become a problem.
Okay. Seeing another question on here for you, what types of normalization methods support on Frida?
So right now, Frida only handles data in terms of presence absence and does not attempt to do normalization. We did write a paper recently about potential data normalization, statistical data normalization approaches for FTICR data. And I'd be happy to, if whoever asks the question wants to reach out to me, I can point you to that paper and where that code lives. But as we get more and more confident about normalization methods actually doing what we hope that they should be doing for statistical purposes, we'll start incorporating that into Frida. But I don't think, I'm not sure the community is there yet, and I would point you to this paper as kind of evidence of that.
All right, Yuri, I can see the codes in GitHub, but there is no code for visualization. If we want to generate plots with output from CoreMS, how can we do it?
Okay, so there's two answers for that question. Like the primary source of generation of visualization from the CoreMS is Frida, right? So the idea is that you have the output from the CoreMS, you can import in Frida and work with different visualizations. But inside the framework itself, like when you have a mass spectrum object, right, you always have functions like plot, like, you know, it's just called that function inside the mass spectrum object. You're going to have a plot of the mass spectrum. And we do that for all the objects inside the framework. And there's also like a class called heteroatom classes. If you want to go back to classic plot, like carbon number versus GBE, or, you know, just basic von Kraeveling diagrams, you can import that class past the mass spectrum object or the LC object into that, and that will generate a bunch of those classical diagrams that you guys are used to look at.
Okay. All right. Will, peak picking of Orbitrap data is performed on reduced profile data or full profile?
So the examples shown here were reduced profile, as that's the default way the instruments are operating. We do support full profile processing because we support any type of data, essentially. You can import transients if you have those and do the full signal processing, or you can import literally an XY table of just data points and load that in as a spectrum object, or you can import these reduced profile data sets.
All right. Is there support for proteomics workflows, Yuri?
The answer is no. We don't have a workflow for proteomics data, but all the signal processing and part of the workflow can be used on the CoreMS, but we don't have a specific workflow for protein identifications. So.
Okay. And Lisa, are there tools available to support statistical analysis and visualization of other types of metabolomics data? I'm tripping over my words. Hopefully you understood what I was saying.
Yeah. Yeah. So we have, as I mentioned, you know, Frida is really a part of this larger suite of tools, and that we're calling MAP, the Multi-Omics Analysis Portal. And one of those other tools is called PMART. And that, you know, it's a similar philosophy in that all of these data have some massaging and treatment that need to happen. But that PMART app, which I'd be happy to provide a link to anyone who just shoots me a quick email to try that out as well, can be used to process GC and LC data. As you know, someone asked a question about normalization. We feel much more comfortable knowing how to statistically normalize GC and LC data from a statistics standpoint than we do FTICR right now. And so those types of capabilities are built into the PMART module.
And Will, how does the accuracy of CoreMS assignments compare to other software tools?
So it's a tough question to answer because accuracy presumes you have a known truth. And oftentimes working with these samples, and we're, again, assuming what the truth is, and we're assuming that the best data we can get on the best instruments is the most likely to be correct. But doing a sort of a like-for-like comparison between tools is can be tricky because you have to make this assumption that one of them is true or that one of them is more true. In sort of anecdotal comparisons, we tend to find that when you're limited to CHO space, most of these tools give you the same assignments. But when you start including these heteroatoms, we find that some tools, because of how they're structured and coded, actually give you different results depending on the order of operations you perform the search in. Whereas with CoreMS, it's at least robust to the fact that it doesn't matter necessarily which order you perform operations in, you'll get a consistent result. So in that regards, I would argue that it's more accurate, but in terms of a number, it's impossible to say.
And Yuri, does CoreMS support absorption mode FTMS processing?
No, it doesn't. it only works on magnitude mode FG, but I want to stress that, this is an open source tool, software, and we would love, to see the community embracing it, and maybe, adding new functionalities like, absorption mode FG for signal processing. That would be awesome.
Okay, we're going to end with a question for Lisa. Are you planning on using Reticulate to interface between Freda and CoreMS, or is there another strategy you're using?
So we're primarily using MinIO to basically just grab files as they're available, but we are using Reticulate and Celery and some of these other back-end options for handling our stack here. But I think Yuri can correct me if I'm wrong, that the primary mechanism is MinIO to grab files from CoreMS.
Yes, it is.
All right, well, with that, we are at the top of the hour. We want to thank you for joining us today, and also encourage you to reach out to Yuri and Lisa and Will if you have additional questions. If we didn't get to your question today, I will make sure that the question gets to the speaker that can answer it, and we will be in touch with you. Hope to see you back here in December for the next EMSL webinar. Have a great day.